Bismillahirrahmanirrahim. My name is Kashif Iqbal. I am MPhil scholar in microbiology. Today discuss the topic name is novel insight into immune system of bats. Content list: Introduction, innate immune in bats, pattern recognition receptor and RNA viruses, limited inflammatory response, dependent DNA virus sensing in bats, adaptive immunity in bats, immune cell population in bats, and immune response and virus spillover and conclusion. Introduction: In recent years, viruses similar to those that cause serious disease in human and other mammals have been detected in healthy bats. These including like coronavirus, filovirus, paramox viruses. Coronavirus causes severe acute respiratory syndrome, filovirus causes Ebola virus disease, and paramox virus causes Marburg hemorrhagic fever. The outbreak of viruses like Nipah virus infection in Malaysia and Bangladesh, the Hendra virus infection in Australia, and severe acute respiratory syndrome SARS pandemic of 2003. The severe acute respiratory syndrome SARS pandemic of 2003 laid the foundation for investigation into bats mammal of the order Chiroptera. During the SARS pandemic of 2003, there are 8,098 people were infected and 774 people are died. These viruses do not appear to cause disease in bats, and this has led to multiple studies exploring the ability of bats to harbor these viruses with no observed clinical consequence. The order Chiroptera is diverse and consists of over 300 species of our bats. Chiroptera consists of two suborders. The first one is Yemtrochiroptera, which comprises megabats, and second one is young Chiroptera, young Chiroptera, comprising all remaining macrobats families. Within these suborder, bat species display tremendous diversity in size, morphology, diets, social interaction, and ecological niche. In this review, we summarize major development in the understanding of this novel. First one is bat antiviral response, highlighting unique property of bat immune system and comparing and contrasting antiviral signaling pathways in bats and human cell. First of innate immunity in bat cell. Mammalian cells have evolved conserved pattern recognition receptor that sense pathogen association molecular pattern PAMPs derived from virus, bacteria, and parasites. Following viruses infection, infect cell initiate signaling event that induce the expression of antiviral and pro-inflammatory cytokines. Like antiviral cytokines such as interferons activate the expression of interferon stimulating genes that inhibit virus replication through different mechanisms. Next one is the availability of whole genome and transcriptome sequence for some bats species as enabled in silico data mining to detect homologous of the mammalian innate immune system. Approximately 3.5 of the transcribed genes identified in the black flying fox are thought to be immune related. In the closely related Jamaican fruit bat, 466 immune related genes have been identified by transcriptome analysis, and 2.75% of genes, roughly approximately 407 genes in the Egypt fruit bat are immune related. The genome and transcriptome of at least 18 bat species are currently available in database, providing important insight into the evolution of their immune system and antiviral immunity. Below we discuss the evolution of antiviral response in the bats in the context of cellular detection of RNA and DNA viruses. First one is bats pattern recognition receptor and RNA viruses. Pattern recognition receptors such as toll-like receptors are eventually conserved across the animal kingdom. Due to the importance of bats as a reservoir of zoonotic RNA viruses, there is particular interest in identifying the intracellular pattern recognition receptor in bats cells that may engage antiviral signaling pathway following infection with RNA virus. In human cell, endosomal toll-like receptor 3 and 7 and 8 detect viral RNAs. Full-length transcripts for toll-like receptor 1 to toll-like receptor 10 have been sequenced in Petrus Electo, and toll-like receptor 13 pseudogenes has been detected, but their function in bats have not been fully characterized. The role of toll-like receptor 3 in sensing exogenous double-stranded RNA has been confirmed in the cell for the big brown bats, but interaction studies and identification of ligand binding domain in bats toll-like receptor 3 have not been performed. Computational structural analysis of toll-like receptor 8 sequence from 21 bats species have been identified. Different between bat and human sequence. 63% of bat toll-like receptor 8 genes have evolved under purifying selection, and 7% amino acid sequence that make up the ligand binding domain of toll-like receptor 8 different between bat and other mammalian toll-like receptor 8 of protein. Cytosolic pattern recognition receptors such as retinoic acid inducible genes and melanoma differentiation association genes 5 that detect exogenous RNA in human cell and have been detected in most bat genomes. Retinoic acid inducible genes and melanoma differentiation associated genes 5 from P. electo have similar primary structure and pattern of tissue expression compared to their human counterparts. Similar to human and rodent cell, P. electo kidney cell produce interferons in response to stimulation with the poly-IC. Limited inflammatory response. Pattern recognition receptor signaling through the adaptor protein to express antiviral and pro-inflammatory cytokines. Regulation of such inflammatory response is critical in order to limit tissue damage. Many severe virus infections are associated with excessive inflammation associated pathology in humans. Bats have evolved novel mechanism to limit virus-induced pro-inflammatory response while maintaining type 1 interferon response to limit virus propagation. Like one, this is a infected cell and this is neighboring cell are affected by infected cell. These are infection spread in cytosol of the cell. Like first one, step by step are discussed. Bat cell mount antiviral response to RNA virus but limit the expression of inflammatory cytokines. Infection with RNA virus such as the Cinda virus या transfecting cell with single stranded RNA is detected by toll-like receptor 3 to 8. Like this is the RNA virus. and cytosolic receptor retinoic acid inducible gene 1 and melanoma differentiation associated protein 5. Activation of these receptors activate downstream adapter proteins such as a mitochondrial antiviral signaling protein like this. This is a RNA virus. Double stranded RNA virus activate the retinoic acid inducible gene and MDA genes, which activate the MAVS mitochondrial signaling antiviral signaling protein. Adapter protein activates cellular kinase and such as tank bending kinase 1 protein, which is turn activate interferon regulatory factor 3 या interferon regulatory factor 7 and nuclear factor copper light chain enhancer of activate B cell stimulate the expression of interferon such as interferon alpha, beta, mega, kappa and lambda. Pro-inflammatory cytokines such as interleukin 8, TNF alpha and interleukin 1 beta respectively. Like this toll-like receptor 7, 8 and 3 activate the adapter and cellular kinase. Mitochondrial antiviral सिग्नल प्रोटीन जो है वो भी क्या करेंगी, इसको एक्टिवेट करेंगी सेलुलर काइनेज को. Then interferon bind to interferon alpha beta receptors 1, 2 and interferon receptor 2 on infected and neighboring cell to activate the JAK state signaling pathway kinase such as a genus kinase and tyrosine kinase 2 that phosphorylatory signal transducer and activator of transcription protein like this. Interferon 3 cellular kinase which activate the neighboring cell stat signaling like this and phosphorylated stat protein stat 1 and stat 2 merge with interferon interrelated